about Megasporophyll. Last class we have discussed about the male reproductive unit present in the flower, that is Microsporophyll or stamen, and how the stamen develops, how the pollen grains develop, how the pollen grain will cause fertilization. All the things related to that we have discussed. So let us discuss in today's class the female reproductive unit in the flower in details. So Megasporophyll is commonly known as the carpel. Alright, and you know that carpel in the form of a hole would be known as gynoecium. Right? Now, regarding gynoecium, we can say that gynoecium consists of one or many carpel or pistil, isn't it? That's why there are two conditions that might arise from here, which you have also learned in the morphology chapter. One carpel means monocarpellary condition, whereas many carpel will mean multicarpellary condition. Monocarpellary and multicarpellary. Okay. Multicarpellary condition can be again of two types. Alright. That is apocarpus or syncarpus. Apocarpus means free carpel, like for example Mycelia, whereas syncarpus condition means fused carpel, example being papaver. Alright? Are you able to recall? All these terminologies were already discussed in the morphology chapter. Now, regarding the structure of the carpel, you know very well that the structure of the carpel consists of three parts: stigma, style and ovary. Stigma is the site where pollen grains would lodge during fertilization. Style is the path through which the pollen tube would pass through, or it would transfer, right, carrying the two male gametes. And ovary is the globular base of the carpel, within which remains the ovule, which is otherwise technically known as megasporangium. Do remember this. Megasporophyll means carpel and megasporangium means ovule. Alright? Now, next let us discuss the structure of ovule in details. Now this also has been discussed. See, this was the structure all about. Right? Now, the ovule or the megasporangium, it consists of a stalk called the funicule. Funicule is the part or the stalk of the ovule with which it remains attached to the placenta within the ovary. Alright? Now, next you can see this joining. The joining that or the attachment point of the funicle and the body of the ovule. This is called hilum. Right? The point of attachment of the funicle with the body of the ovule is called hilum. Now, if we look at the body of the ovule, the first thing you can see is that the body of the ovule is being surrounded by certain protective layers. These are called integuments. So we have an outer integument. Similarly, we have inner integument. Right? Internal to that remains a mass of parenchyma tissue that's called the nucellus. Alright? It's called the nucellus. Now, as you can see, in the whole body of the ovule, there is one section which is not being surrounded by the integument. This is going to be called as micropyle. Alright? And the opposite end of the micropyle will be termed as salazar. Now, regarding the integument or nucellus, we can differentiate the ovule. Like, first let us take the consideration of integument. Based on the number of integuments, the ovules can be of three types. Number of integuments is the criteria here. Alright, so accordingly we have three conditions that can occur. First is unitegmic, means one integument. Alright, example can be higher dicots. Alright, the next possibility is bitegmic, which means two integument. Example here would be primitive dicots and monocots. Alright, likewise ategmic. That would mean no integument. For this, the example I am writing here, that is Santalum and Lorenthus. Alright, so these are the important examples that you should remember. Alright, based on the number of integuments, we can differentiate the plants into unitegmic, one integument, bitegmic, two integument, and of course ategmic without an integument. Examples are important. Now, next category is on the basis of nucellus. Alright. On the basis of nucellus, we can have two types of ovule. First, crassinucleate, that is well developed or thick nucellus. Alright, and the second one, tenuinucellate, that is thin or poorly developed. Alright, example would be here polypetally, here gamopetally. Alright, these being the structure of ovule. Now, according to the orientation of the funicle or the body of the ovules can be of 6 types. Let us discuss them. Types of ovule. So there are 6 types of ovule. First, orthotropous. Orthotropous means erect ovule. Alright, here the structure is going to be as such. So as you can see, funicle, salazar and micropyle are at a straight line. So this is called a erect or orthotropous ovule. Example is all gymnosperms or among the angiosperms, polygonum. Alright. Next, anatropous. Anatropous ovule means inverted ovule. Alright, here the micropyle will be tilted towards the funicle and salazar will be on the opposite end. So this is inverted ovule. Example would be 82% of all angiosperms. Alright, then hemitropous. Hemitropous ovule means half inverted. Means it will be halfway between orthotropous and anatropous. So it would be as such. Here, micropyle and salazar are perpendicular to the funicle. Alright, example is going to be ranunculus. Alright. Fourth, campylotropis. Campylotropis means curved body of the ovule. The body of the ovule will be having a slight curvature like this. Alright, there will be a slight curvature that is going to be opposite to the funicle. Alright, example would be mustard. Alright, now the fifth one, let me draw here. Fifth one is amphitropus. Amphitropus means body plus embryo sac shows curvature. That means the body as well as the embryo sac both would be showing curvature. There in Campylotropis, the body was showing curvature, but there was no curvature in the embryo sac. In this case, embryo sac will be so curved that it would take up a horseshoe shape. Alright, so it would look like this. So the body is also curved and even the embryo sac is becoming curved and it is taking up a shape like this. Horseshoe shape. Alright. Example would be poppy. Alright, and the last one, Sarsinotropis. Here, the funicle will be circularly surrounding the micropyle. It's going to be like this. These being the funicle, see it is circularly surrounding the micropyle. This is the micropyle, right? So this is called Sarsinotropis. Example would be Opuntia. So hope I have been able to make everything clear to you all people. This is for today's class. In the next class we will start with Megasporogenesis, where we learn how a well-developed embryo sac will be formed inside the ovule. Thank you.